Okay, we're here. I've been picking on this aisle lately for the joke. Who's got a joke before we have a couple of announcements? Matt, you provided a joke, haven't you?
That you in my email. I sent a joke.
Oh, I've forgotten it. So
No, can we cut that one? It's
I'm,
not very good. I mean, it's okay.
no,
I
no,
can, I can.
yeah, give it a shot.
For my brother. All right. So, how do you spot a blind man at a nudist beach?
How do you spot a blind man at a nudist beach?
It's not hard.
Of course, I knew what you were referring to, but I didn't anticipate the punchline. That's not a bad joke at all. Did did you leave out the punchline? And I was supposed to. Oh, okay. Well, okay. That's good. Okay, couple of announcements. Number one, before I forget, uh, do recall that the second midterm is this coming weekend, starting on Friday. For those of you who want to be traveling on Saturday, I'll make it available at 6 in the morning on Friday. It's the same format as you had before. It will cover chapters 3, 6, and 11. I think that's the order we've been doing them. I'm a little bit concerned about whether I'm going to get through as much of 11 as I want to test you on. So I'm going to skip the Kahoot on gender. But I say first, any questions about the test? Okay, take it on D2L whenever you like. Once you start, you got 90 minutes, and you got to be finished before midnight Sunday night. Good. Okay, so I've pretty much wrapped up what I had to say about gender, and I've got one more sort of update. But any um, any questions about anything I raised before? Okay, two updates. One is because someone raised this, and I wanted to talk about an example. Someone asked um, if there are some countries where homosexual behavior is illegal, but trans isn't. And in fact, in the government of Iran, for the last several years, there's where homosexuality is totally illegal. There's definitely a whole effort to get those people to become trans, because see, if a guy grows up and he's attracted to men when he grows up, that's a problem. But if he then converts and becomes a woman and loves men, then that's not a problem, right? So there is this push pull between ways of dealing with one's sexual orientation, right? And until lately, in our culture, uh, the way people dealt with the homosexual orientation was either they suppressed it, sometimes for their whole lives, or they took on the role of a gay man or woman. Another way to go is for those individuals to and to become trans, and therefore they can express their sexual behavior in what is seen as a heterosexual way. And what's totally unknown is, you know, how many of the people who become trans do so because they're trying to reconcile who they're attracted to with what society says they're supposed to be. And there's really no good way to get a handle on that. Finally, as all this kind of a recap, I'll remind you that there are some trans women where, in fact, they're attracted to women. So in autogynephilia, those individuals are, um, attracted to women, and yet still they feel this need to change their gender and go to, sometimes a lot of trouble and take quite a bit of risks in making that change because it's that important to them. Questions about any of that? Just today, I got an update on some data came in. Remember, I told you that right now in the United States, for some time now, there's been a growing cohort of people that identify as trans, that the number of people who identify as trans, while still, very much the minority, has been rising. But there's a latest set of data from the latest cohort reaching adulthood where the rate of trans identity has been cut in half. Now, that could just be, a wiggle in the data. Um, and it doesn't mean that it'll continue to decline, but there is some evidence that the number of people who by adulthood consider themselves to be trans may have peaked and may be about to decline. So I just put that out there because I don't think anyone knows. But you can be thinking about, and I think it's pretty obvious to think that there might be political debates going on that are surely having an effect on how likely people are to identify as trans and how likely they are to carry through with that want that need to change their gender. How's that? So I think that there's a distinction that's an important one that we're going to get at today. We've been talking about what's your gender identity and what's your sexual orientation. And remember, I've given you some terminology to divorce those two, to say, I don't know what your gender is, but if you're attracted to men, you're androphilic. And if you're attracted to women, you're gynophilic. And we've seen that those are not necessarily always joined. They can work independently. And here's what I'll propose to you as a fundamental dichotomy in sexual behavior. And I'm going to start it off with a poll of you. So, how many of you remember your first crush? The first time you saw someone, maybe on TV, in movies, on the playground, somewhere, where first you felt different about that person, right? Okay. So, how many of you were under 10 years old when that happened? Look at that, the majority of people. Several interesting things about that. Number one, it's well before puberty. So it's nothing to do with pubertal hormones. Number two, how many of you were younger than 6? Almost no, yeah, a few. That's it. Always, always precocious. I was pretty much 6 years old, and it was seeing Marilyn Monroe on television. How stereotypical. Uh, I am just so damn white and so damn straight. It's really embarrassing. Now, think about that. And how many are there any of you where whatever the gender of that first crush, you are now sexually attracted to the opposite sex? Well, no, no, no. Who you find attractive? Yeah. See? I mean, how many genders there are? That's influenced by and by the way, that experience with that's what virtually everybody says. And I want you to think about that first crush. It is totally strange, right? It is not that you had that sexual orientation at birth because you were somewhere between 6 and 10. Most people were. So you'd already been in the world 6 years or more, and you weren't having crushes on anybody. You weren't following social rules about who you are. You might have had a girlfriend, boyfriend, might have pretended to get married and stuff like that, but you didn't have that feeling. So there's something weird about that. And we don't have a term for it, but I want to point out to you, and I want you to think about this, that this was a decision that your brain made, and you were not consciously aware of how it came to that decision. You were not consciously aware of what input went into the decision, and you are not consciously able to overcome it. And in an era where, thank goodness, well before you, where gay people were condemned because they were choosing a lifestyle. Well, who chose which sex you're attracted to? Nobody. Not gay people any more than straight people. And I'll go a bit further. Even today, whatever gender you're attracted to, you have no control over who you find sexually attractive. I mean, what you decide to do about your sexual attraction, that's totally within your control. But you can't choose any more than you can choose which art you find attractive or which music you find attractive. These are things over which you have no conscious awareness of the decision being made and over which you have no conscious control. And I'm going to propose to you that that's totally different than what we've been talking about a bit about genders, where clearly gender is influenced to some extent by social influences. Otherwise, why would the number of trans people be changing, right? Going up, going down. Why would there be some genders with some societies with more than two genders and some with only two? And potentially one day a society with four or five different genders, right? So there's this fundamental divide. And what I'm going to try to convince you of is that this mysterious feeling that you have is that there are, I'm not saying that they are only influenced by biological factors. I don't think that means anything. But we're going to see they are influenced by things that happened before you were born. One of the reasons why you had no choice in the matter. So you see where we're going? Question? Good. It's a great question, and everyone can answer it for themselves. I personally don't think you can choose to find opera great and Billy Joel horrible, right? I mean, you can tell people you love opera and you hate Billy Joel. You can tell people, boy, that person's really attractive, even if you're not attracted to them. But that's not what you're talking about, right? No, I don't think we have any. I'm not. Are any of you aware of being able to decide who you're going to find sexually attractive? Ah, never have. But believe me, it can be incredibly inconvenient not to be in charge of that. And yeah, okay. So to talk about this, we're going to talk about quite a bit of about experiments in animals. Let's see. I wonder what happens if I do that. So here are some of the learning objectives. We'll get there. In animals, there's sexual behavior, and I'm showing you this picture of rats copulating because this is going to provide us with a lot of background material that is interesting. This is the male. So when you put male and female rats together, if the female is an estrus, which is to say she is is about to ovulate and therefore is very fertile, you put them together. The male, whatever animal you put in a male's cage, he will always sniff around and try mounting it a few times. And the mounting it consists of coming up from behind, grabbing the flanks with his forelegs, and rhythmically pushing his pelvis against the backside of the animal. If it's a male, nothing happens. The male acts like nothing's happening, and pretty soon the male that mounted will give up. If it's a female and she's not an estrus, she'll act like nothing's happening. The male may try several times, and then he'll give up. If she's an estrus, when he gives her that same stimuli, she has this reflexive response known as lordosis, where she raises her head, she lifts her rump, if she's a rodent that has a tail, she'll push it to one side. And all of this makes it easier for him to get his penis inside her vagina when he does that rhythmic pelvic thrusting thing. So it's known as lordosis, and it's a very sensitive measure of how receptive the female is. So we're talking about the female's sexual receptivity. And there's been a lot of experiments done with this behavior. I can tell you that if the female's been primed with hormones so that she'll show lordosis response, she doesn't care who provides the stimuli, because I can take my two forefingers and put them on her flank and push my thumb against her rump, and she'll lift her rump, lift her head, and push her tail to the side, even though I know she's unhappy with me being close to her at all. And that's why I'm calling it a reflex. It is reflexive. But for us, the important point is that it's gated by hormones. So if you remove a female's ovaries, she'll never show this behavior again. But if you remove her ovaries and then give her hormones, gonadal hormones that replicate the normal ovulatory cycle, give her estrogen injection one morning, estrogen injection the next morning, and then give an injection of progesterone the third morning. About 6 hours after that last injection, now she will show lordosis, either in response to a male who mounts her or in response to me and my thumb. And so we say that she's reached receptivity. And it requires gonadal steroids. Which particular gonadal steroids varies from species to species. In rats, it's got to be estrogen for a couple of days, followed by progesterone. In other species, estrogen alone is enough, et cetera. And we don't need to care about that. What's interesting is you can give those same hormones, estrogen for 2 days, then progesterone, to a male rat, and he never shows lordosis, even if another male gets up behind him and mounts. He shows no interest in showing that behavior. So something is different about the nervous system of males and females, so that when you give females those hormones, they show the spinal reflex, and when you give males those same hormones, they pay no attention. And it was in 1959 that this group, and this is like one of the few times when I want you to remember the names of some authors, Phoenix, Goy, Giral, and Young. They published a paper in 1959 where they were working with guinea pigs. And in guinea pigs, as with rats, females will show lordosis if given estrogen than progesterone. And males, given those same hormones, will not. And the experiment they did is in these guinea pigs, they treated pregnant guinea pigs. Oh, so that's all the stuff I just told you. In this experiment, they treated pregnant guinea pigs with testosterone. Now, guinea pigs have very long gestational period, unlike rats, right? So guinea pigs, when they're born, I don't know if you know this, they already have fur and they have teeth, and they don't have to nurse. They can eat soft food, right? So they have a long gestation period. But when they gave testosterone to the mother, the dam, remember that's a female of a species that's being used for breeding. When they gave the testosterone to the dam, it went across the placenta just fine, and all of her offspring were exposed to testosterone. And so when they did that, they found that when those females grew up, if their brains were exposed to testosterone early in life, they would not show lordosis. Somehow, what happened to them before they were born was going to influence their response to hormones in adulthood and change their sexual behavior. And they proposed what came to be known as the organizational hypothesis, because it had just been worked out how hormones organize the phenotype of males and females in sexual differentiation. And they said, well, just as for the body, genetic sex determines gonadal sex, and gonadal sex uses hormones to determine phenotypic sex, right? The Y chromosome causes testes to develop, the testes releases testosterone, penis and scrotum, et cetera. In the absence of a Y chromosome, ovaries form, there's no testosterone, and a female phenotype occurs as well. That had just been worked out, maybe by 1952. And they said, maybe the brain is just one more organ that is listening for that signal. And there's something sort of that is that when the brain is exposed to testosterone early in life, that exposure organizes the brain, changes its structure permanently, and makes the animal show more masculine behavior. Everyone follow that so far? And in the absence of testosterone, as happens in normal females, then the brain is organized in a female fashion. And that the behavior is just an output to let you know whether the brain differentiated in masculine or feminine fashion. How's that so far? And if you think about it, there's something elegant about that solution in that there's one factor, it's either there or it's not. And then both the body and the brain gets organized to show the behavior that's most likely to result in reproduction and offspring. Okay? Yeah. Nick. Ask the question again. What? Yeah. Collective about their mates. Have that as well as that. Well, so I mean, you're absolutely right. In many species, females will absolutely have a preference which male they will accept. In these studies, there's no chance for that to happen. The the male is in a 20-gallon aquarium with sawdust, and you drop the female on top of him, and she's got nowhere to go. She's not able to make a choice. And probably, rats, it's not clear how much the female gets to choose. What is interesting is that if you give the female someplace where she can go hide, what's interesting is every time she will go hide some of the time after each intermission, but she comes back. And even though she's able to stay away, she'll, so she's interested in mating too. And she might change the timing of it. Men are always in a hurry, right? Um, but but in these experiments, she's not given any chance to exert a choice of her partner. Great question, but they purposely kept that from being an issue in these experiments. So here's some of the experiments that got done then. So you take an untreated female, put him with a male who mounts him, and he won't show lordosis, even if you give them those hormones. And in turn, he will mount receptive females. Untreated female, it's the converse. What they found was if you castrate males at birth in these species, rats, mice, many other species, when they grow up, if you now give them hormones, they'll show lordosis, which they would not have shown before. And they're much less interested in mounting female. And here we're seeing the experiment we just talked about, where you give testosterone around the time of birth to females. And when they grow up, they won't show the lordosis, even if you give those ovarian hormones. And instead, they're much more interested in mounting other females. So what they were proposing is that steroids act on behavior in two fundamentally different ways. One is the way we talked about with the adult female, where ovarian steroids regulate whether the female is going to show sexual receptivity. Or when we talked about males, when you castrate males in a wide variety of species, pretty soon they stop mounting. And if you give them testosterone, they start mounting again. So these are examples of hormones having an activational effect. They activate the nervous system temporarily and increase the likelihood of mating behaviors. But Phoenix, Goy-Giral, and Young said there's a second, totally different way that hormones affect mating behavior, and that is when they act early in life to permanently alter the structure of the brain and therefore permanently masculinize behavior. How's that? And it turns out what happens after they publish that 1959 paper is thousands of papers get published where they manipulate these things, manipulate hormones early in life, and then study sexual behavior late in life. And when you do that, almost always, you can make the animal's behavior as masculine or feminine as you want, just by controlling how much testosterone they see early in life and how many gonadal steroids they get in adulthood. So you got to organize the system first, and then you use hormones to activate it. Yeah. Well, I mean, it's not, you can't really, compare timelines in rats versus humans, but it's in both species, the peak there is a peak of testosterone before birth in virtually every mammalian species. There's a peak of testosterone right at birth and less thereafter. After that, the details differ. In guinea pigs, the peak is found like in the middle of gestation. In rats and mice, it comes towards the end of gestation. In humans, it happens when? Early in gestation, right? First trimester. So eventually, people did experiments like this in lots of different species. So in monkeys too. Monkeys mount for all sorts of reasons, but males mount for sexual reasons. And there's a very specific way they clasp the hind legs of their partner and thrust, etc. And female monkeys will only show this behavior when they're hormonally primed. And what people found was that if you gave female monkeys testosterone before birth, they were much more likely to show these sorts of behaviors in adulthood. And let me remind you that testosterone, like all steroids, the half-life once you inject it into an animal is very, very short. In these experiments where they inject testosterone, it's all gone within 24 hours. So it's not that it's hanging around to keep acting. So somehow the hormone acts now to change behavior weeks, months, years later. And the organizational hypothesis was meant as a way of explaining that. And, you know, it would eventually people would, there's some behaviors that only listen to adult hormones, right? So turns out yawning in monkeys. By the way, does anyone know why animals yawn? All across mammals, they yawn. Anyone know why? What function it serves? Nobody knows. Totally unknown. What we know is it serves as a social signal. In some primates, where the males have great big canine teeth, it's definitely, a display of, look how big my teeth are, don't mess with me. And in monkeys, you take an animal, whatever their sex, and you give them testosterone today as an adult, and they'll show more yawning. And that's kind of the exception to the rule. That is, where you don't have to give hormone early in life to see a masculine behavior in adult life. You okay so far? There's one complication that I feel like I have to talk about, which is there's this weird business in rats and mice, which are the most studied species. I've already mentioned to you that testosterone serves as a precursor to the formation of estrogens. So in fact, once a molecule of testosterone gets made, there's an enzyme that very rapidly converts it to estradiol. This enzyme is called aromatase because if you take an organic chemistry, you can see this carbon ring becomes aromatized. And I'm not going to talk about why. Yeah. And organic compounds that have an aromatic ring like that, they tend to have odors associated. You tend to be able to smell it. That's why they got that name. Anyway, so if you could make estrogens airborne, you'd smell them, and testosterone you wouldn't. And I don't know why I went down that pathway. They're too heavy. They don't become airborne. Okay. Um, so the ovary is chock full of aromatase. So any testosterone precursors that get made in the ovary, virtually always they get converted to estrogens. And another place where there's chock full of aromatase is the placenta. Any androgen that happens to go into the placenta will get converted to estrogen before it comes out again. So it turns out that in many species, including most of the rodents, the masculinizing effect of androgen on the brain isn't acting on androgen receptors, it's acting on estrogen receptors. So to understand that, here's how it works. In rodents, Testosterone is made by testes. It gets to the brain and enters cells there. You'll remember steroids being very lipophilic. They pass into membranes easily without a membrane-bound receptor. In target cells, there's plenty of aromatase that converts the testosterone to estrogen. The estrogen then binds to estrogen receptors, not androgen receptors. And it's the estrogen receptor complex that then alters gene expression and in rodents will cause the brain to behave in a more masculine fashion. And several species, rodents, ferrets, several other species, that's how testosterone masculinizes the brain. This might not be what it was originally seen as paradoxical, right? Because people were thinking the old-fashioned way that there are male hormones and there are female hormones. But here's a great example of how there's no such thing. All gonads make all the steroids, and in some species, natural selection has tapped estrogen receptors to masculinize the brain, and in others, not. Having told you about that, because there's such a great literature about it, I want to go back and say that if gonadal steroids masculinize the human brain, we know it's not doing so through aromatization because for lots of reasons, some of which we'll come to later. But I need you to understand the mechanisms of aromatization before I take Abder. So what happens then to the estrogens that mom makes? I've told you before, every pregnant woman is teeming with estrogens, right? And so why aren't female rats masculinized by the estrogen they got from mom? That'd be disaster. Every animal acts like males, and that'd be the end of the species. Well, it turns out that in rodents, there's a circulating protein called alpha-fetoprotein, and it binds in rodents, it binds to estrogens. So any estrogen from mom is sequestered by alpha-fetoprotein, and therefore the female fetus's brain isn't masculinized. When you inject testosterone, you short circuit that defense. When you inject testosterone, it's not bound by alpha-fetoprotein. The testosterone goes to the brain, gets converted to estrogen outside the reach of alpha-fetoprotein, and then masculinizes the system. Everyone see how that works? Sometimes this is referred to as the aromatization hypothesis, because for a while, people were having a hard time believing it could happen, but it clearly does in rodents and other species, but not in primates. Okay? Sexual behavior of rats that have been exposed to testosterone early in life or rats that have been shielded from testosterone early in life. On the D2L page, go to the video about 60 Minutes. So a long time ago now, 60 Minutes did an episode where they were talking about things. And guess who they went to as someone who knows how to castrate male rats at birth and have them grow up, et cetera. So you'll get to see a male rat who lost his testes at birth. And when he grew up and was given those ovarian hormones, he showed beautiful lordosis behavior on camera, on cue. There was no chance he wasn't going to do that because that experiment's been done so many times, okay? And in the meantime, you'll see someone who more or less resembles me, who is strangely younger than I am now. I don't know how this happens. Okay, so for a long time after 1959, after the organizational hypothesis made a big stir, and I will say in terms of the sociology of science, the results they got, other people had gotten those same results before, had seen those same results, but what they did is they offered a coherent framework to explain it that suddenly made it easy to understand and easy to remember. If the brain is exposed to testosterone early in life, they'll grow up to be more masculine and less feminine. And if the brain isn't exposed to testosterone early in life, they'll grow up to be more feminine and less masculine. So it was easy to remember, and suddenly it explained a whole bunch of behavior. If you've taken molecular biology, a similar thing happened when they finally found out the code, the three uh, amino acid codons that, pardon me, the three nucleotide codons that code for a particular amino acid, et cetera, when what had been a confusing field of different enzymes that do different things, and everyone's trying to work out all these, suddenly it became simple. DNA makes RNA, and RNA makes protein. You can remember that. But more importantly, it explained an enormous amount of data that had otherwise been hard to organize and think about. And so that was what was true of the organizational hypothesis as well. And from the very beginning, they proposed that this was going to affect the structure of the brain, but it would be, I don't know, 20 years or more. It would be, yeah, almost 20 years before anybody ever looked to see if there were sex differences in the structure of the brain. And the reason they didn't look is because they thought they'd be hard to see. Okay. But it turned out that once someone looked, they were really huge. So, get this out of the way. So in 1976, some people published a paper where they looked at the brains of birds, zebra finches and canaries, and they had been working out which parts of the brain are important for birdsong because they knew that males sing and females don't. And so they had mapped out which brain regions were important for that. And when they looked at some of the sections, they noticed something, that there were these brain regions that they knew were important for birdsong that were much larger in male birds than they were in female birds. And this difference was so large, if you had the stained brain section on a glass slide, you could look up and see it with your naked eye, and you could see, oh, that's from a male and that's from a female. Nobody expected the differences to be that prominent, but they were. And eventually, at UCLA, a postdoc listened to a talk about birdsong and thought, well, that's cool that there's sexual dimorphisms in the brains of birdsong, but that's not important because birds aren't an important species. We only care about mammals. And of course, for an anatomist of that time, the most important mammal was rats. No, I know, not humans, but rats, because they're easy to study. So he went to the lab and he looked at rats and he knew exactly where to look if you wanted to find a sex difference in the brain. It was the preoptic area, because tons of research had shown that the preoptic area is absolutely crucial for ovulation and it's absolutely crucial for male sexual behavior. Across a wide species of mammalian species, if you lesion the preoptic area, males stop copulating. They will not copulate. So, okay, he had some slides in the lab that were from the brain of the preoptic area, and he looked, and what he saw was that if in males there was this dense staining nucleus in the preoptic area that was a lot bigger in males than in females. And so it quickly became called the sexually dimorphic nucleus of the preoptic area. And again, it was so um, obvious that if you held the stained slide up to the light, you could sex it. Oh, that came from a male, that came from a female. To make it a little personal, in 1977, when they discovered this, it wasn't published yet, but I happened to be taking neuroanatomy at UCLA, the graduate neuroanatomy. And a guy, Roger Gorski, was supposed to come talk to us about neuroendocrinology. But instead, he comes to us and he has all these slides and he makes us hold the slides up to the light and see, oh, that dot's a lot bigger in that one. That must be from a male, or the dot's a lot smaller in that one. That must be from a female. And then the floodgates opened. People started looking at all different parts of the brain, and it was really impressive how many you could have published a journal that was just publishing articles about sexual dimorphisms in the brains of animal X in region Y. I mean, they were everywhere. And we'll talk some more about this nucleus, where you could also find sex dimorphisms in the spinal cord. So it turns out in the lumbar spinal cord, these dense staining cells, these are motor neurons, and they send their axons out of the spinal cord, out the ventral root, and down the pudendal nerve, and innervate the bulbocavernosus muscle, also sometimes known as bulbospongiosus muscle. And once I worked out where these motor neurons were, that they were in this weird position in the midline, then noticed that when I looked in females, there were hardly any motor neurons there. And so I called this the spinal nucleus of the bulbocavernosus. And again, the sex difference was so prominent, you didn't need a microscope to see it. So we'll be talking more about the SDNPOA and the SNB in a moment. Questions about that? And the one theme all of these sexual dimorphisms share is this. They follow the organizational hypothesis beautifully. You can make the nucleus as masculine or feminine as you like just by controlling how much testosterone the animal sees. And almost always, it's got to be how much testosterone they see early in life, usually just before or just after birth. So here was this anatomical signature that told you how right the organizational hypothesis was. The other thing they all have in common, almost all of them, is that the sexual dimorphism, once it's programmed at birth, you can't manipulate it in adulthood. Giving hormones or taking them away in adulthood, for most of these cases, does not affect the sex difference. Questions about that? Let's have some examples first. Don't know why I have it in that order. First, let's talk about this slide. So Roger Gorski then and his colleagues, they saw that in unmanipulated males, they had a large SDNPOA in adulthood. Oh, and I should tell you, the SDN portion of the POA is just a minority of the preoptic area. In fact, the SDNPOA in rats, and for that matter in people, is about the size of a big grain of sand. So these are really tiny, right? So it's not the whole POA, it's that portion of the POA. Anyway, and when they looked in females, the same nucleus was much smaller. If they gave a female testosterone early in life, they would have a large SDNPOA in adulthood. But if they waited until adulthood and gave her testosterone, it had absolutely no influence on the structure of this nucleus. How's that? So in rodents, the SDNPOA obeyed beautifully the organizational hypothesis and the aromatization hypothesis, because it turns out the SDNPOA of rodents is masculinized by estrogenic metabolites of testosterone that act on estrogen receptors, which is very suggestive because that's the same mechanism that alters the behavior of these animals to masculinize them or not. How's that? I'll go back to the S&B just to talk to you that of all the sexual dimorphism in the nervous system, and maybe this isn't surprising, it's the one in the spinal cord that we understand best. It's pretty straightforward. So it turns out in rats, before birth, the motor neurons of the SNB form, send their axons out, and innervate the bulbocavernosus muscle. And in rats and mice, before birth, both sexes have a bulbocavernosus muscle. And the SNB motor neurons innervate them in both cases. And what happens next is about the time of birth, in females, the muscles die. And once the muscles die, the motor neurons die. In males, the muscles don't die, they get bigger. And that's because testosterone keeps the muscles from dying. So in this case, testosterone, when it masculinizes the periphery, causes a masculinization of the nervous system. And we even know that what the testosterone is doing is causing this muscle, number one, to live, and number two, to give a chemical signal called a trophic factor to tell the motor neurons don't die. And so, if in this female, if we give her a single injection of testosterone early in life, just before birth is best, then the muscle does not die, the motor neurons do not die, and she'll have a masculine spinal cord the rest of her life. So in this case, we understand what's critical about the critical period for masculinizing the brain. You have to get the testosterone there before the muscles and motor neurons die. If you wait until they're already dead, the testosterone can't masculinize the system. Nick? Ah, great. No, clearly not for all muscles of the body. It's not even true for all species. So in humans, again, before birth, the bulbospongiosis cavernosus muscle looks the same in males and females. It doesn't die in human females because it's making the constrictor vestibuli, the one that closes the opening of the vagina. But it does grow a lot larger in male fetuses than in female fetuses. And indeed, the spinal cord nucleus in humans, there's more of those motor neurons in males than there are in females. And it's almost certainly the same mechanism because the period of motor neuron death that happens in humans coincides with the period of testosterone secretion by males. It's a great question. Even in rats, this is a direct action of androgen receptors. So if you look at animals that have the androgen receptor disabled, basically animal models of androgen insensitivity syndrome, they have a totally feminine S&B system. Without androgen receptors in the muscle, it will die. So it is not the result of aromatization, unlike what happens in the brain of rodents. Another reason to talk about the SDN POA is because eventually people asked whether there's an SDN portion of the POA in humans. And I won't, bother you with all the trivia about who discovered what, when, and where. But indeed, when people looked in the preoptic area of humans, Gorski declared that there were 4 different nuclei in the SDN portion, and that 2 of them were sexually dimorphic. And he named these nuclei, what a horrible decision. He named them the interstitial nuclei of the anterior hypothalamus. Ugliest name ever in neuroanatomy. At least he didn't call it Gorski 1, 2, 3, and 4. And he numbered them. And he said 2 and 3 are sexually dimorphic, but 1 and 4 aren't. I don't need you to remember any of that. But people tended to zero in on INA 3. So we'll sometimes call it INA 3, and then we'll sometimes call it the SDN POA. So it is about 1.5 times larger in men than it is in women. So how did that come about? I want to hear 2 hypotheses about how it is that men have a larger INA 3 than women. One is testosterone, that the men brains were exposed to testosterone early in life. I guess I didn't tell you, in rats, the testosterone, the way it makes the SDN POA bigger in males and females is it keeps neurons from dying in males. And so males end up with more of them. And again, if you give the testosterone after the period of death is over, it's too late to rescue them. So one possibility is that prenatal testosterone organizes the human SDN into a masculine configuration. What's another hypothesis? There's only one. Yeah. But another possibility is that the different social experiences of boys and girls mean that their INA3 will have a different development, right? Entirely possible. It's even sort of hinted. So in rats, the SDNPOA is 3 to 5 times larger in volume in males than it is in females. But I just told you, INA3 in humans, it's only 1.5 times larger in males than in females. And I'm sparing you all the other sexual dimorphisms that people described in animals, but I will tell you that almost always when they looked in humans, they could find a sexual dimorphism there. But the interesting thing is the sexual dimorphism in humans is always smaller in proportion than it is in animals. And I don't know if I'll get around. Eventually, people found one nucleus that's larger in female rats than in males, called the AVPV. And when they looked in humans, sure enough, in females, the AVPV is larger than in men. And again, in rats at least, the AVPV is larger in females because in that nucleus, testosterone causes more neurons to die. And we'll come back to INA3 later because this is the nucleus that Simon LeVay found is, yes, larger in men than in women, but is smaller in gay men than it is in straight men. So we'll come back to talk about this later. But one reason to think that Maya's alternative hypothesis could be going on, oh, I didn't mean to put that there, but is eventually people looked at brains from people of lots of different ages, including prenatal specimens. Because this nucleus is so small, you can only see it post-mortem. You cannot detect it with MRI or CAT scans or anything like that. But, people die all the time. And so in one study, they looked at the number of cells in the SDNPOA from birth until, they had a few people that were 100 years old. And it's a very interesting pattern. The convention is triangles are male, because they have a pointy part, and circles are females, because they have a hole. I didn't make it up. It just happened. And you can see that in the first 5 to 10 years of life, there is no visible sex difference in INA3, the SDNPOA. And what we know is during this period, the boys and girls are living in different worlds in terms of how they get treated, how people talk to them, what things like that. And so it's entirely possible that the difference in INA3 that unfolds at 10 years of age was caused by social factors. And this is just something where it's a slightly different number and it's been, altered to make it smoother. By the way, the other thing that's interesting is that in both sexes, INA3 shrinks as you get older. And this is a little more shrinkage than, here, enjoy your time. Your brain size is going to asymptote at about early 20s, okay? And it's going to stay very stable until you reach 40 years of age. And starting at 40, every 10 years, your brain is going to shrink about 5%. So, you'd have to live an awfully long time for it to shrink and go away altogether. But that's just the nature of things. And so probably this decline in old age is probably just reflecting that. Then there's an experiment that I like that I want to tell you about that talks about the possibility of social influences affecting sexual dimorphism in the nervous system. So in this experiment, a researcher, Celia Moore, she was studying rat maternal behavior. And I'll tell you something about rat maternal behavior that you probably don't know. One of the things the mother does, you probably know, of course, the mother normally nurses the pups so they can feed. But another thing she does is she's grooming them all the time. And it's especially important that she groom their anogenital region because rats at birth cannot defecate or urinate unless there's some sort of stimulation happening of their anogenital region. So mom, every day, several times a day, she picks up each pup, licks the anogenital region, and that stimulates urination and defecation. And by the way, mom doesn't spit out any of that stuff. She just ingests it all. And there's a really cool system where the metabolites that are coming out in the feces of her pups is going to inform the composition of her milk as they get older. And, you know, we're not going to worry ourselves about that so much. But if she doesn't do that, the pups can't live no matter how much she feeds them, right? And what Celia Moore noticed is that some rat pups are much better mothers than others. That some rat dams are much better mothers than others. And that what she noticed was that all rat dams, they lick their sons more often than their daughters. And that's an interesting observation by itself. Number one, it means they can tell what sex each pup is. Nobody knows how exactly, but it might be based on the metabolites in the urine, right? Because the males will have some androgen metabolites that the females don't. We don't know. But she always licks her son's anogenital regions more than her daughter's. And then she thought, well, maybe they can tell them apart because of the smell. So she made it so that the dams couldn't smell for a while. We say that she made them anosmic. And what happens when she does that is she stops licking her males more than her females, and she licks everybody less. So then she asked about the S&B system of males, and it's a complicated graph, but the bottom line is this. If those males who grew up with a mother that didn't lick them very often didn't have as many S&B motor neurons survive as those who had their moms lick them a lot. I don't think anybody saw that coming. It had seemed to be all worked out. Testosterone keeps the muscles alive, and the muscles keep the motor neurons alive. What's mom got to do with it, right? Supposedly nothing. But what she showed was that one of the things that has to happen for as many motor neurons as possible to survive is that the pups have to receive anogenital stimulation early in life. And without that, not as many motor neurons survive. She didn't measure the target muscles, but probably that's being affected too. So here's my question for you. Is this a biological effect or a social effect? Someone choose and tell us why it is. Yeah, what do you think? Is it a biological effect or a social effect? This stimulation of the anogenital region that keeps motor neurons alive. What do you think, Zach?
I say social because the force driving it is an external force, not an internal force.
The external force being mom.
Yeah, it's looking coming from an external source, not
So
the rat itself.
you're absolutely right. Clearly, this is a social effect. Okay, absolutely correct. And it can't be a biological. It's absolutely, if there's not a mom there to do it, it doesn't happen. Who wants to take the other side? That it's a biological effect. Okay, thanks, Maya. You think it's a biological effect. Tell us why.
Sure.
So in the interest of arguing for this, because
Oh, you're committed.
okay, I'm committed. I believe this. All right, it's biological. Because there is like maybe there's like like the stimulation keeps it. So like just causing stimulation.
The tactile stimuli of mom licking the anogenital region. Yeah.
Yeah,
And that's a biological effect.
totally.
No
And then
doubt.
the mother's but then the mother like sniffs it and has this like instinct because if she doesn't
No, stop. No, don't go any further.
Okay.
Okay, it's clearly biological because it's affecting sensory fiber firing, right? That's biological as it get. And it's influencing cell death. How can you be more biological than that? And Maya, you're absolutely right. It is absolutely a biological effect, not a social effect. Oh, wait a minute. Uh, you can't tell these. There's no such thing as a social effect or a biological effect anytime, ever. And this is a great illustration of that. And that idea that there are social influences and they have nothing to do with biological and vice versa, it's one of the most pervasive ways of thinking in our society. And it's totally wrongheaded. You cannot understand anything if you think that things can be divided that way. Think about this. Any influence an experience has on an individual that's going to change their later behavior, that has to have some influence on the brain, right? It's got to change the number of synapses, the flavor of synapses, the number of cells. There's no way for an experience to alter behavior unless it has a biological effect. That is, it changes biological variables. Likewise, any biological influence, a hormone, a tumor, etc., if it's going to change behavior, right? It is going to, if it is going to be important, it's going to change the animal's behavior. So it's both biological and it's behavioral. And there's no possibility of having a behavior that's only biological or only social. There is no difference between genes and experience. There is no difference between hormones and stress or hormones and trauma, right? If they're going to affect behavior in either case, then they're going to affect biological variables and they're going to have an influence on behavioral variables. So, this next slide, if you're looking at it, because in all of this, the question is, what has this got to do with people? Are these same hormones having any influence on human behavior and sex differences in behavior? And the answer is maybe. But here's one way, and we'll be talking about this next time. But here's one way in which I'm absolutely certain testosterone has a masculinizing effect on the human brain. It's inescapable. This has to be the case. If we think about rats, the only reason mom licks the sons more is because they have testosterone. So the only reason she gives them that extra attention is because she's picking up on a biological signal. Oh, this is a son. I'm going to lick him more. And therefore, he'll have more motor neurons in his spinal cord, and therefore, maybe he'll give me more grandchildren. Like that. So here's one way testosterone masculinizes the human brain. The testosterone acts on the periphery and causes a penis to develop. And as soon as the baby is born, it comes out. And as soon as individuals know that it's a male, they start treating it differently. And so one way testosterone masculinizes the human brain is that it causes a penis to develop, and then people change their social influence to alter the individual's behavior. And so it's another way where I'm trying to get you to see there's no distinction between biological influences and social influences. They go hand in hand. But as a result of that, I want to refine the question. The question isn't whether testosterone has a role in causing sex differences in human behavior. The question is testosterone having an influence on sex differences in human behavior because of something that testosterone does to the brain before birth, or because of something testosterone does to all the people around the individual after they're born? Or then I'm going to pare it down just a little bit more than that. The question is, are there any sex differences in human behavior or any sex differences in the human brain that are caused because testosterone before birth acts on the brain? And finally, another way of saying it. The question isn't the question, that question can be boiled down to, are there any prenatal influences on sex differences in human behavior? Because whatever happens before birth, whatever it is, it cannot be socially mediated because nobody knows what the sex is. They haven't had a chance to treat the individual differently based on what testosterone did to the crotch, right? And therefore, if we really want to know whether there are hormonal influences on human behavior, what we really need to ask is, are there any prenatal influences on human behavior? And if they are, they can't be attributed to differences in gender treatment because nobody treats the babies differently before they're born based on nobody can tell whether they're male or female before they're born, and therefore they don't have a chance to influence the process. Questions about that? So, in prep for next time, I need to have a short seminar about statistics. So, hopefully you know that almost every biological variable is distributed in what's known as a normal fashion. So make this any biological variable you want on the x-axis. And here we're keeping track of how many individuals have that score. In psychology, you've seen this in terms of IQ scores, right? Where there's a distribution like this, but it can be any biological variable. And if I was teaching statistics, as I have done at other times in my life, we'd have a long discussion about how is it that every biological variable is always organized or always distributed in this bell-shaped curve. But I'm going to spare you that. I just want to say that we can come up with statistical terms to describe every standard distribution, also known as a normal distribution, which is to say any biological variable. There's a mean, the average, and typically the average is also the most common score. And if we now talk about standard deviation, it turns out that virtually all individuals will be within 3 or 4 standard deviations. What's a standard deviation? The standard deviation can be summarized as the average deviation individuals are from the mean. So we don't care whether it's above the mean or below the mean, but on average, there'll be an average of how far away each individual is from the mean. So take every individual score, calculate how far away it is from the mean, and then average those together, but don't pay any attention to whether they're bigger or greater than the mean. That's the standard deviation. And we can abbreviate it as sigma. And across these, no matter what the variable, it'll turn out about 68% of the individuals will be within 1 standard deviation of the mean. And if we expand that to another standard deviation from the mean, we'll add another 27% to the total. And if we look at the area under the curve, that's proportional to what proportion of the population will have those scores. In psychology, statistical tricks are used to make sure the mean is always 100, and the standard deviation is always 15 IQ points. And that's just done with little mathematical manipulation. But it still is true that 68% of the population will be within one standard deviation, and et cetera. They'll be distributed this way. And I don't need you to remember any of where the distributions are. I just want you to understand the idea. And the reason I'm telling you this is we can talk about a way of gauging how big a difference is, how big a sex difference is between two populations, let's say male and female. And we do that with something that's known as D prime. D prime is how many standard deviations different are the two populations. Here, how many standard deviations difference is there between the behavior of men versus women? And if the difference is huge, it'll be like 3 standard deviations. But there are no sex differences in human behavior that get anywhere near that. In fact, I'll tell you about a sex difference in human behavior, a D prime, that you know about, which is the sex difference in adult height in humans. Doesn't matter what the measure is, doesn't matter what you look at, it'll turn out that that's a D prime of about 2, meaning the means of males and females is 2 standard deviations apart. And no one has ever found any, well, that's not true. Okay. And so we can gauge how big a sex difference in behavior is by what's the D prime? How many standard deviations apart are the two populations? And what we're going to see is there are very few behaviors that are that different in men versus women. But this is going to be a useful statistic for us. Okay, so last chance for a question about normal distributions, standard deviations, means, D primes. We'll take this up again next time. So see you Thursday.